constructed cloning vector or an artificial cloning vector, artificial plasmid pBR322. The name pBR322 conforms with the standard rule for vector nomenclature. Of course, P stands for plasmid, BR stands for Bolivar and Rodrigues, the two researchers who developed pBR322. And 322 distinguishes this plasmid from others developed in the same laboratory. Of course, there are other plasmids, pBR325, 327, 328, etc. So I repeat the slide. pBR322 constructed cloning vector. The name pBR322 conforms with the standard rule for vector nomenclature. P indicates that it is indeed a plasmid. BR stands for Bolivar and Rodrigues, the two researchers who developed pBR322. And 322 distinguishes this plasmid from other developed in the same laboratory. There are also plasmids called pBR325, pBR327, pBR328, etc., which was constructed in the same lab. So they are Bolivar and Rodrigues. This artificial vector was constructed by them in the year 1977. So it is one of the most popular and commercially used early purpose built plasmid based vector system. So the first vector system, artificially constructed vector system that is used up to date for gene cloning experiment is pBR322, which was constructed by Bolivar and Rodrigues in the year 1977. So if you look into this diagram, so this is the structure of pBR322, which is an artificially constructed cloning vector. The size of pBR322 is 4363 base pair or 4.3 kilobase. It has an origin of replication to autonomously replicate. It has two resistant genes as the selectable marker, the ampicillin resistant and tetracycline resistant gene. Ampicillin resistant gene has site for restriction endonuclease, including SCA1, PVU1 and PST1. On the other hand, tetracycline resistancy can be cleaved with the help of restriction endonuclease, HIND3, BAMH1 and SAL1. Ampicillin and tetracycline resistant gene can be separated with the restriction endonuclease EcoR1. So this is the structure of the constructed cloning vector pBR322. I repeat, pBR322 is the smallest constructed cloning vector of 4.3 kilobase size of 4363 base pairs. It has two resistant gene as selectable marker, ampicillin resistant gene and tetracycline resistant gene. Ampicillin resistant gene can be restricted with the restriction endonuclease SCA1, PVU1 and PST1. On the other hand, tetracycline resistant gene can be cleaved with the restriction endonuclease HIND3, BAMH1 and SAL1. It also possesses EcoR1 between ampicillin and tetracycline resistant gene. The vector has an origin of replication to autonomously replicate. Regarding the pedigree of pBR322, how was it constructed or where are these fragments taken from? The plasmid was in fact designed in such a way that the final construct would possess these desirable properties. The ampicillin resistant gene is originally resided on the plasmid R1 from Escherichia coli. So R1 plasmid is a typical antibiotic resistant plasmid which is present in natural populations of Escherichia coli. So the ampicillin resistant gene, which is there on the left hand side of pBR322, as you see the picture, is from a typically antibiotic resistant plasmid in the natural populations of Escherichia coli. I repeat the sentence. The ampicillin resistant gene originally resided on the plasmid R1, a typically antibiotic resistant plasmid that occurs in natural population of Escherichia coli. On the second hand, if you see the tetracycline resistant gene is derived from the plasmid R65, a second antibiotic resistant population of Escherichia coli. So I repeat, the tetracycline resistant gene is derived from R65 plasmid, a second antibiotic resistant plasmid. And of course, the replication, the origin of replication of pBR322, which directs for its multiplication in the host, is from PMB1, which is an collagen producing plasmid called even of Escherichia coli. So, மூணு different plasmidல இருந்து எடுத்து construct பண்ணப்பட்ட vector is pBR322. Ampicillin resistant gene is from R1 plasmid. Tetracycline resistancy is from R65 plasmid, and origin of replication is from PMB1 plasmid. So that is what is this slide all about. I repeat the pedigree. Pedigree of pBR322 is very important to mark a question. The plasmid was in fact designed in such a way that the final construct would possess these desirable properties. The ampicillin resistant gene originally resided on the plasmid R1, a typical antibiotic resistant plasmid that occurs in natural population of Escherichia coli. The tetracycline resistant gene is derived from R65, a second antibiotic resistant plasmid. The replication origin of pBR322, which directs multiplication of the vector in the host cell, is originally from PMB1, which is closely related to the collagen producing plasmid called E1. So this is the diagram that depicts the construction or the pedigree of pBR322. So in the left-hand side top, you have R1. On the right-hand side top, you have R65. R1 shows ampicillin resistancy. R65 shows tetracycline resistancy. Finally, the origin of replication, if you see, is from PMB1. You can see in the diagram PMB1 beneath. So we have come across with various stages of constructing a vector. pBR312 was first constructed with the help of ampicillin resistance and tetracycline resistance gene on origin of replication. Then it is rearranged to construct a vector pBR313, and the size fragmentation was reduced to 4363 base pair in pBR322. So if you look into the last down, you find pBR322 with ampicillin resistancy and tetracycline resistancy gene with origin of replication. So the origins of replication, the diagram B, if you see, ampicillin resistancy is from R1, tetracycline resistance is from R65, and origin of replication is from PMB1. So pBR322 is a constructed cloning vector which is constructed by removing fragments from 3 different plasmids and putting it together to produce an chimeric DNA. So what happens with the normal Escherichia coli cell and E. coli cell, which is having pBR322 plasmid, or which is transformed with pBR322 plasmid, is this diagram. Since we are using 2 different antibiotic, ampicillin and tetracycline, we call them as selectable markers in this case. And what we do, if you see, when you incorporate the plate with the ampicillin and tetracycline, or the combination of both, because one plasmid has 2, you can also implement it with ampicillin, or you can go it with tetracycline, or both you can incorporate in the medium. So you're taking an agar medium, you're adding to it 40 microgram per ml ampicillin or 15 microgram per ml tetracycline, or a combination of both. The normal E. coli cell, go to the top left, the normal E. coli cell, of course the E. coli cell doesn't have anything inside, but it does have chromosomal DNA, which is not mentioned there. So normal E. coli cell, there is no plasmid, so it cannot survive because it is sensitive to these 2 antibiotics. On the other hand, the E. coli cell, which is transformed with pBR322, will have resistance to ampicillin and tetracycline, thereby producing the colony in plate that is incorporated with ampicillin and tetracycline. That is how you select the Escherichia coli transformed cell with the pBR322 type plasmid. Since these 2 antibiotic resistant gene is used to detect the recombinant clone, you call them as selectable markers. What are the useful properties of pBr322? Concentrate with utmost care. The first useful feature of pBr322 is its size. A cloning vector ought to be less than 10 kilobase in size to avoid problems such as DNA breakdown during purification. pBr322 is 4363 base pair, which means that not only the vector itself be purified with ease, but so can the recombinant DNA molecule constructed with it. Even with 6 kilobase of additional DNA, a recombinant pBr322 molecule is still, of course, a manageable size. I believe you understand this sentence. So the molecule is smaller in size. So in addition, you can go in with 6 kb, still it will be of manageable size. I repeat the sentence. The first useful property of constructing pBr322 is because of the size of the molecule. A cloning vector ought to be less than 10 kb in size to avoid problems such as DNA breakdown during purification. pBr322 is 4363 base pair, which means that not only can the vector itself be purified with ease, but so can recombinant DNA molecule constructed with it. Even with 6 kb of additional DNA, a recombinant pBr322 molecule is still a manageable size. The second feature of pBr322 is that it carries 2 sets of antibiotic resistance gene. As I already told you, it contains either ampicillin or tetracycline resistancy as selectable marker. And each marker gene includes unique restriction sites that can be used in cloning experiment. Insertion of new DNA into pBr322 that has been restricted with PST1, PVU1 and SCA1 inactivates the ampicillin resistant gene, and insertion using BAMH1 Hind3 SAL1 inactivates tetracycline resistant gene. What do I mean is, in case if you are having 3 different restriction endonuclease, PST, PVU, SCA1, if you're going to cut, it will inactivate ampicillin resistancy. Whereas, in case you have 3 other different restriction endonuclease, BAMH1 Hind3 SAL1, you're going to restrict it, then it will inactivate tetracycline resistant gene. This great variety of restriction sites that can be used for insertional inactivation means that pBr322 can be used to clone DNA fragments with any of several kinds of sticky. And so many different molecules can be used to cut the the vector system. So you can use several restriction enzymes to produce the sticky-ended structure. I repeat the sentence again. The second feature of pBr322 is that it carries 2 sets of antibiotic resistance gene. Either ampicillin or tetracycline resistance can be used as selectable marker for cells containing the plasmid, and each marker gene includes unique restriction sites that can be used in gene cloning experiments. Insertion of new DNA into pBr322 that has been restricted with PST1, PVU1 or SCA1 inactivates the ampicillin resistant gene. On the other hand, insertion using BAMH1 or Hind3 or SAL1 inactivates tetracycline resistance. This great variety of restriction sites that can be used for insertional inactivation means that pBr322 can be used to clone DNA fragments with any different several kinds of sticky-ended molecule. The third advantage, if you see, is it is a reasonably high copy number plasmid. So the third advantage of pBr322 is that it has a reasonably high copy number. Generally, there are about 15 molecules present in a transformed E. coli cell, but this number can be increased up to 1,000 to 3,000 by the technique plasmid amplification in the presence of a protein synthesis inhibitor such as chloramphenicol. And E. coli culture therefore provides a good yield of recombinant pBr322 molecules. So I repeat the sentence again regarding the third advantage. It is reasonably a high copy number plasmid. Generally, there are about 15 molecules present in a transformed E. coli cell, but this number can be increased from 1,000 to 3,000 times by plasmid amplification in the presence of protein synthesis inhibitors such as chloramphenicol. An E. coli culture, therefore, provides a good yield of recombinant pBR322 molecules. So basics are these three different advantages. The size, regarding the selectable marker it possesses for insertional inactivation, two different selectable markers. Number 3 is its copy number. This is one summary of the salient features of pBR322. So size is less than kb10, so less than 10 kb in size. 2AD gene, that is antibiotic resistant gene. It is having unique MCS. So many different restriction enzymes can be used to cleave. The phenomenon insertional inactivation can be well brought about, and it is a high copy number plasmid, basically has 15 molecules per cell that can be increased to 1,000 to 3,000 molecules by using a protein synthesis inhibitor, chloramphenicol. What is plasmid amplification? Preparation of plasmid DNA can be hindered by the fact that plasmids make up only a small proportion of the total DNA in a bacterial cell. The yield of DNA from a bacterial culture may therefore be disappointingly low. Plasmid amplification offers a means of increasing this yield, so increasing the copy number. The aim of amplification is to increase the copy number of a plasmid. Some multi-copy plasmids, those with copy number of 20 or more, have the useful property of being able to replicate in the absence of protein synthesis. This contrasts with the main bacterial chromosome, which cannot replicate under these conditions. So as I told you earlier, multi-copy number plasmid, there are a variety of plasmids that can multiply in the absence of protein synthesis, albeit which cannot be done with the help of bacterial chromosomal DNA. Bacterial chromosomal DNA requires protein synthesis for its replication. So that is what is going to be used for plasmid amplification. This property can be utilized during the growth of a bacterial culture for plasmid DNA purification. After a satisfactory cell density has been reached, an inhibitor of protein synthesis, example chloramphenicol, is added and the culture is incubated for a further period of 12 hours. During this time, the plasmid molecules will continue to replicate even though chromosome replication and cell divisions are blocked because of inhibition of protein synthesis. The result is that plasmid copy numbers of several thousands may be attained. Amplification is therefore a very efficient way of increasing the yield of multi-copy number plasmid. So if you look into this diagram below, on the left-hand side, you have the bacteria with bacterial chromosomal DNA and black-colored multi-copy plasmid. For your visualization, it is colored black, multi-copy plasmid. The copy number is 20 plus. You are incubating it in the presence of chloramphenicol. Since there is no protein synthesis or inhibition of protein synthesis takes place, the chromosomal DNA does not replicate, the cell does not divide, whereas the copy number of the plasmid increases to 1,000 plus. So that is what is the diagram below. And this technique of attaining increase in the copy number is called as plasmid amplification. Now, what is insertional inactivation of antibiotic resistant gene? The process of inserting a foreign gene and inactivating an pre-existing functional gene. Now go into the diagram A. You have the top A I'm talking about. Okay, so you have normal vector molecule, you have a blue colored molecule which has a pink color region of your gene product and the target gene for insertional inactivation. So the normal vector will produce the product. Now go to the B, recombinant vector. molecule. So you have a vector with an interrupted, disturbed target gene. So the gene that produces the product is no longer functional. It is being inactivated. So no gene product is produced. And this phenomenon is called as insertional inactivation. What happens with pBr322? Come to the left, figure 5.7, the cloning vector pBr322. On the A, the normal vector molecule, if you see, you have ampicillin resistant gene and tetracycline resistant gene. As it was mentioned earlier, you have restriction site for various restriction endonuclease onto the either region, the ampicillin and tetracycline. One example which is depicted is BAMH1. Now, tetracycline resistant gene has restriction site for BAMH1. So the normal vector molecule is ampicillin resistant and tetracycline resistant. In case if you are inserting a gene by cleaving the molecule with the help of BAMH1, the tetracycline resistant gene that shows resistance to the antibiotic tetracycline, it's interrupted, whereby the target new gene is inserted so that the gene remains inactivated. Go to the diagram B. A recombinant molecule containing an extra piece of DNA inserted into an BAMH1 site. So the recombinant pBr322 molecule, if you see, you have the vector, pink colored ampicillin resistant gene for your visualization. On the other hand, you have tetracycline resistant gene, which was green in color, interrupted with the new DNA molecule, which is gray in color. So when the tetracycline resistant gene is intervened or interrupted or inserted with a new DNA insert, restricting it with an BAMH1 site, the tetracycline resistant gene is no more functional. Tetracycline is converted to tetracycline sensitive. So now it is designated down as ampicillin resistant and tetracycline sensitive. And this phenomenon of inserting a foreign gene and inactivating the pre-existing functional gene is referred to as insertional inactivation. Insertional inactivation always does not imply antibiotic resistant gene. There are various genes that can be interrupted for the product revealing. So insertional inactivation in case of pBr322 is with antibiotic resistant gene. So in insertional inactivation of antibiotic resistant gene in pBr322. How are recombinant molecules selected with pBr322? If you see, insertional inactivation of an antibiotic resistant gene pBr322 has several unique restriction sites that can be used to open up the vector before insertion of a new DNA fragment. Here, one example is given, BAMH1, for example, cuts pBr322 at just one position within the cluster of gene that codes for resistance to tetracycline. A recombinant pBr322 molecule, one that carries an extra piece of DNA in the BAMH1 site, is no longer able to confer tetracycline resistance on its host, as one of the necessary genes is now disrupted by the inserted DNA molecule. Cells containing this recombinant pBr322 molecule are still resistant to ampicillin, but they are sensitive to tetracycline. That is why it is designated ampicillin resistant and tetracycline sensitive. So this is the diagram or the position of BAMH1 site in pBr322 molecule. Now, how are we going to select? So we are going to use a technique called as replica plating technique, which you have already studied. You are going to plate a medium with ampicillin, a plate with tetracycline, and of course, you're going to identify the colony. Go to the topmost diagram. You have colonies or recombinant colonies onto the, it is not recombinant and non-recombinant colonies in the ampicillin medium, because the organism, if it is recombinant or non-recombinant, will be resistant to ampicillin. Okay, now you are going to what is called as replica plating. You are going to prepare replica plating in two different plates. You're going to grow the organism in plate containing ampicillin and in a plate containing tetracycline. So ampicillin plates, all the type of organisms will grow. Tetracycline plates, imagine a situation, only what is called as non-recombinants will grow. The reason is recombinant, you have cut to with the BAMH1, tetracycline is no more functional, tetracycline resistancy cannot be shown, the medium is incorporated with tetracycline, the organism has become sensitive, it cannot grow. You will incubate and then select for the position of the colony. Ampicillin resistant tetracycline colonies grow on a tetracycline medium, can be located with the first master plate. Come to the left hand side for the description. Screening for pBR322 recombinants is performed in the following way. After transformation, the cells are plated onto ampicillin medium and incubated until the colonies appear. All of these colonies are transformants. Remember, untransformed cells are ampicillin sensitive and do not produce colonies on the selected medium. What do I mean is, pBR322 is an artificial plasmid. Number transformation pannirundha mattume host cell it will grow. இப்போ டிரான்ஸ்ஃபர்மேஷனே நடக்கல ஹோஸ்ட் செல்லுக்குள்ளேயே அது போகலன்னா அந்த ஆர்கானிசமால வளர முடியாது. ஆம்பிசிலினும் இருக்காது இல்ல. That is the meaning. But only a few contain recombinant pBR322 molecule. Most contain the normal self-ligated plasmid. So self-ligation நடந்திருந்தது அப்படின்னா வளரக்கூடிய வாய்ப்பு உண்டு. To identify the recombinants, the colonies are replica plated onto agar medium that contains tetra